morning, everyone.
Good morning.
My name is Tawei Ma, and today I will be talking about tissue engineering the geometry of 3D cardiac structures that beat on shaped biocompatible matrices. So first I would like to mention some things about the heart. The heart is the hardest working muscle in the body. It is constantly contracting and pumping blood. For this reason, the heart does not have time to take a break and recover when damage is done. This inability makes the heart vulnerable to contractile dysfunction, and which may lead to heart failure. Traditional methods for sustaining critical heart function often involve surgical procedures. What if there was a way to provide the heart with a patch, similar to a Band-Aid, comprised of a person's own cells, and that would promote healing after its application? This is where alternative methods involving biocompatible systems would help address this issue by providing a more proficient and less invasive system that will restore damaged cardiac tissue without altering natural biological function. So, in summer of 2014, my group and I experimented in this area of research. P19-CO6 cells in tissue culture were differentiated into beating cardiac tissue using 1% DMSO on a variety of matrix materials, including fibrin. Several tissue culture surfaces were used as substrates for matrix construction. We know from the literature that DMSO promotes cardiogenesis through inhibition of the Wnt signal transduction pathway. Fibrin is a protein that's involved in blood flow. Ultimately, we lay down fibrin, differentiated cardiac cells on it, and a few weeks later, beating monolayers were produced. This is what our tissue culture plates look like. Large beating areas such as this one were found throughout the plates, with some areas beating in synchrony. These results were great as it relates to our fibrin patch idea for the heart. However, others are also producing similar beating monolayers. So we took this idea a step further and asked, why not shape the fibrin matrix and see what happens? And so we did. So how did we do this? When making our fibrin matrix, we manipulated the fibrinogen from the center of the tissue culture plates and shaped it using a simple technique. Dipping into the fibrinogen from the center of the plate wells to the outer edges of the plate before adding the thrombin, which would polymerize the matrix. The higher viscosity of the fibrinogen allowed for an easy manipulation and distribution of the product. Thrombin was then added, coating the set fibrinogen and resulting as the fibrin matrix. From this technique, we were able to produce some interesting suspended contracting structures. Here's an example of such a structure. We call this one Battlestar Galactica. Here's another example, Bermuda Triangle. The most interesting structures, however, were also the most simple. Here's an example of a single beam strand in which you can see striated patterns, a characteristic of cardiac tissue. A closer look reveals the individual cardiac cells coating the fibrin strand. And this is how it looks like under a microscope through an HD monitor. we were able to produce single beating strands. Imagine these strands having a hollow structure and being able to transport blood or other fluids throughout the body. The concept here is that these fibrin strands could be shaped more precisely with the use of electrospinning, 3D printing, and other technologies. These strands could be manufactured to include hollow cores in various diameters, ranging in different shapes and sizes. As I mentioned before, these strands could serve as vessels in the body for replacement of damaged arteries and other fluid pathways. Now, let's take a moment to go back and talk about our cell differentiating factors. In addition to DMSO, we experience with other factors on their own and in combination as well. Of the ones tested, DMSO provided the best results in differentiating the P19-CL6 line into cardiomyocytes. Another factor worth mentioning is C59. C59 was consistent with the degradation of the fibrin matrix. As demonstrated here, the fibrin matrix would begin to diminish until completely eradicated. What this tells us is that C59 may play a role in the reduction of removal of scar tissue from damaged areas in the heart or other organs. So to get an idea how C59 and DMSO might perform if used in direct treatment, a safety study was conducted in conjunction to this one. C. elegans were used in this safety study. The pharynx of the C. elegans was examined using fluorescence microscopy. The pharynx in this case closely resembles the heart, as its purpose is to circulate blood of the worm and simultaneously functions as a feeding structure. This safety study confirmed the toxicity of DMSO, resulting in adverse effects. C59, however, showed no noticeable effects. Our overall results: the pluripotent cell line P19-CL6 was differentiated to form being cardiac structures on a fibrin matrix by classic DMSO induction. Several types of cardiac structures were shaped through the manipulation of fibrin configuration. C59 induced the degradation of the fibrin matrix. C. elegans were used to investigate the safety of the small molecule inhibitors used in this study, confirming the toxicity of DMSO while showing no adverse effects from C59. So the final concept: a fibrin patch as well as artificial arteries could potentially be shaped into cohesive structures for autologous repair in the human body. DMSO, though toxic, could be used in vitro as a precursor in cultivating cardiac cells. C59's fibrin eradicating properties could potentially be used in vivo for the removal of scar tissue, thereby replacing it with healthy functioning tissue. So for future directions, the initiation of cardiogenesis during development involves an as of yet undefined exact sequence of signaling events. Cardiac cells and other non-beating cell types can easily be derived through an inhibition. The objective of an enriched cardiac cell material containing Purkinje fibers required for coordinated beating remains an elusive goal. 4 areas of interest are feasible to investigate beyond the present experiment. Future experiments will focus on the differentiation of cardiac tissue to pacemaker cells using direct reprogramming due to the introduction of the TBX18 gene. The efficiency of production of cardiac tissue will be explored via modulation of the Notch pathway. it is possible to envision a fiber matrix material embedded with genetic factors for reprogramming cells. Our current aim is to shape the matrix further using magnetic, electrospinning, or 3D printing methods. It is possible that a combination of these methods would enhance our objective of creating a larger coordinating cardiac structures than those demonstrated in this research. Thank you.
Thank you. Any questions?
Yes. Could you go back to that last slide? Okay, so down at the bottom there, you talk about the use of 3D printing methods for actually, to actually develop the matrix.
Yes, so we could potentially use a 3D printing device to make a hollow structure, kind of like in that two-phase cell. So this one right here, it's not hollow, but if it were hollow in the middle, it could potentially be used to pump blood or other fluids in the body.
In one of your earlier slides, are you using bovine tissue cells for this, or human cells?
The P19-CO6 is a mouse derivative. It's from the ovary of a mouse that has cancer.
But you, in one of the slides, you mentioned bovine. The slides.
Oh, wait for the media that we use.
And on that particular one, how are you creating bead?
We differentiate the cells into cardiac cells on the fiber matrix.
So they were electrically beating on their own?
So it
Or were you
started out,
stimulating it in any way?
the P19-CO6 has pluripotent properties, so stem-like properties. So when applied to the plate and through the use of DMSO, they differentiate into cardiac cells. Took about 2 weeks to get them to do anything cool.
How would it be adapted to human health and collaborate with the team itself?
The matrix or the 3D printing?
The matrix.
The matrix? Okay, so the matrix would work as a fiber patch pretty much, sort of like a band-aid. We could potentially use somebody's own cardiac cells and apply them to the patch. And in vitro, cultivate the cells, make the patch, and then in vivo, we would apply the patch to the heart.
Directly?
Yes. So once it's already contracting, it would just be biocompatible with your own cells. That's
And
the
it will
idea.
be feeding out of the bloodstream into the body? Or it's not biological?
It would be adhering to the other cardiac cells in the heart.
But it took 2 weeks to regenerate, to start actually functioning as a
To see
beating
beating signs, yes. And these beating structures were maintained for well over 2.5 months. At 3 months, we called it quits. I'm just kidding. We ran out of media.
Did you consider like the recording action potential of that electrical activity, or
Yes. So
it's actually recording, and you have to people do it outside, you have to use Faraday cage, or
wait, that would be the next step. So in the future studies,
so that's the
I
depends
have that,
what
yes.
to do.
Do you have that picture of the heart?
So this picture to the right shows a heart and the various cells involved So as you can see, there's various types of hearts.
And I could just throw a comment in here. Each of those different cell types has its own EKG. They're all different. And we think of the EKG as being
But then
homogeneous,
the
but it's,
cardiac cells, when they get together, it's
yeah.
electrical,
it's sort of
I think they get
consensus
synchronized,
EKG.
yes?
Right.
Because otherwise that's, they get right in fibrillation. No?
That's a frontier.
Yeah.
area.
Any other questions?
Any other questions?
Do you want to mention the Cedars-Sinai?
So in Cedars-Sinai, they're doing something similar to this idea. They're actually injecting this fibrin glue into the heart directly, which seals the heart, and this fibrin glue contains the person's own cells as well.
When you say to the heart, means to the ventricle or the...
To the site of injury.
Okay.
And it takes 2 weeks before it starts
To be-
synchronizing.
Yes. And these results were consistent throughout the study.
And this is with mice too.
The
Cedars-Sinai.
cell line that we used, the P19CO6.
Cedars-Sinai work is in which model system?
It's in vivo.
It's in human.
Yes.
You had some questions?
Yeah, so it seems like most of your application is related to heart repair.
Yes,
With
and I
this
mentioned that.
application, but is there a possibility or I guess an idea of maybe using this to improve circulation in different areas of the body versus just the heart?
Yes, of course. Major arteries, veins.
Any other questions? If there are no questions, thank you very much.
Thank you.